So Cory Johnson, he's a postdoctoral researcher at the Hiller Lab at MDI Biological Laboratory. As a total aside, MDI Biolab is one of my favorite places in Maine. They just do really extraordinary work. If you haven't had a chance to kind of do a deep dive into what they do, please do. Cory and I are actually, this isn't on here, but this is just an example of how research scientists often do more than just be in the lab and write grants. He and I are on a committee of the Bioscience, we're on the education committee of the Bioscience Association of Maine, which is a trade organization. So many scientists also do all of this kind of support work or public service work to try to encourage different parts of the university. So that's why I get this inside track record of Cory and like, oh, he's going to be really good. No pressure. Cory is interested in the cell biology of kidney vascularization and uses renal organoids and zebrafish to investigate how we may one day be able to rebuild the kidney and develop non-donor solutions to kidney transplantation. And the Huberman Lab podcast is Cory's cultural touchstone. What should we call it that?
Sure.
All right,
great. Yeah. Okay. Thanks very much, Kate. Thanks for the invitation to give a talk here tonight. I'm really excited. I'm doing something totally different, so bear with me. This is why I have notes. So I want you all to imagine a man named John. He's in his early 60s. He eats well, exercises regularly. He goes to visit his doctor for a routine checkup. Everything seems fine, but when the doctor reviews John's blood work, there's a problem. Elevated creatinine levels in the blood indicate his kidneys aren't functioning properly. A follow-up urine test confirms that there's protein leakage, and suddenly, John is diagnosed with chronic kidney disease. Though he's in the early stages now, John eventually faces a sobering reality. He'll likely need a kidney transplant. John is not alone. Nearly 37 million adults in the United States suffer from chronic kidney disease. Of those, nearly 800,000 people have progressed to kidney failure. Only 90,000 patients currently sit on transplant wait lists. Tragically, due to the shortages of donor kidneys, only about 25,000 people a year actually receive one. So, how can we address this critical shortage? Enter the induced pluripotent stem cell, or iPSC. These incredible cells can be generated from your own tissues, like a simple skin or blood sample. And then they can be transformed into virtually any cell type in the body, including kidney cells. Even better, we can assemble these kidney cells into miniature lab-grown kidneys that we call organoids. Kidney organoids not only mimic the structure and functions of real kidneys, but they could potentially serve as transplantable organs in the future that will be personalized to each patient and eliminate rejection risk. But before kidney organoids can move from the lab bench to patients like John, we need to overcome some key hurdles. First, these organoids need to have functional blood vessels. That way they can integrate into the host vascular system, filter blood, and produce urine. This is what I've been working on. Recently in my research, I've discovered that cortisol, a naturally occurring hormone in our bodies and known for managing stress, which I'm definitely feeling right now, can dramatically enhance the formation of blood vessels in these kidney organoids without the need for genetic engineering. Here, let's take a look at what these look like. On the left is the first image of a kidney organoid. This is our control. If you can see in magenta, these are the tubules. These are responsible for nutrient and water reabsorption. You can also see blood vessels in green, and while they do exist in these control organoids, they're pretty sparse. However, when we treat these organoids with cortisol here on the right-hand side, you can still see the magenta tubules, but now you can see an abundant network of green-labeled blood vessels interwoven among these kidney structures. And I actually find these videos quite amazing. Let's zoom in a little bit on some of the structural components that we have in these organoids. Again, we can see in magenta the tubules. In red, we now have these cells called podocytes, which form the functional filtration barrier of the glomerulus in the kidney. If you look closely enough, you can see that the green blood vessels are dipping beneath these podocytes, which indicate they might be actually forming some structural components that would be very interesting for us to see. So we zoomed in on this, and if you can see right there, underneath the red podocytes, there are some small capillary-like structures in green, which is where the arrow is supposed to be. But this is really exciting work. Our ongoing work is focused on refining these organoids. We're currently working to integrate organoids with microfluidic devices that mimic blood flow. This would bridge the gap between lab-grown kidneys and a functional organ. And while significant challenges remain, not all of these being scientific, we're optimistic. And by pushing forward this technology, we hope to revolutionize kidney disease treatment. And one day soon, perhaps John and the millions of others that are affected by chronic kidney disease could receive their own kidney developed by their own induced pluripotent stem cells, and this would eliminate the need for kidney donors completely. Thanks.
Any questions for Cory before I take it over again? Nope. How far along do you think you are between? I know. Everyone asks you this question.
They do.
Sorry. Like from those cool, pretty pictures of little tiny organoids to like, what's the next step? How about that? What's the next step?
Well, the next step is what we're working on. So we're trying to get these organoids set in a way that they actually get blood flow, right? And so without blood flow, the organoids don't mature. And when you don't have a mature organoid, you can't really ask any questions related to disease. So the first step is being able to either integrate them into host vasculature, like a mouse, for example, which people are already doing. But if we can manipulate the organoids using microfluidics instead, that gives us easier access to working with them and changing small things. That's the first step. To answer your bigger question, though, which is what everybody wants to know, how long is it going to take? I think it's going to be on the scale of decades, but I do think it's something that we could eventually see in our lifetime.
And how big are the organoids right now?
Mine?
Any?
Or other?
I mean, just... That took a turn, Cory.
Well... So they range in size, but generally the ones that I grow are a few millimeters in size, but we have a collaborator in the Netherlands who can grow palm-sized organoids, and they've been working on implanting these in pigs.
So that's, I guess, why I was asking the question. If you have larger ones, does that mean you have more room to figure out the blood flow part, or is it like it just gives you more to work with?
Yeah.
And can move it faster?
I think so. Yeah, I think that's a good way to put that. I think having large organoids also creates some challenges because you need flow in order to keep them alive. The problem is these organoids in the very center, they start to get necrotic, which means they die because they don't have access to oxygen and nutrients.
So the images that you showed were super cool. I guess my question is like, having that kind of imaging, so can you talk about what that microscope or what the system was that produced those images? And how many things like that need to be in concert so that you can do what you're doing, right? Like how many different pieces of technology and new science and other breakthroughs help you do what you're doing on the biomedical side?
Sure, yeah. So the microscope that we used is called a Nikon spinning disc. I won't get into the details of it, but it's a confocal microscope, so it takes out of focus light and removes it. This way we can see deeper into the tissue. For the first two images. The last image was taken on our Zeiss 980, which is another confocal microscope, but it does point scanning instead of spinning disc confocal. The larger organoids that people use will use things like light sheet, which we have one at the Institute, but my organoids don't require that. To answer your second question about kind of what resources we need in order to do this, a lot. It's super expensive to grow organoids. And it's 7 days a week. If you can imagine, it's like taking care of a child, right? They require you to change their media every single day. You need to make sure they're alive every single day. And that creates some issues if you don't have enough personnel. And then on top of that, you need reagents and supplies in order to do the actual downstream work to figure out what's going on.
Was there a specific reason for kidneys? And to follow up on that, could this technology be used for other organs?
Absolutely, and it is, to answer that really quickly. So we have another colleague, Mingxia Gu, who's out at UCLA right now. She's working on vascularizing lung and intestinal organoids, doing beautiful work. The reason we chose kidneys is because it's the largest organ transplant that's needed in the United States. So, like I said, there's 90,000 people or so that are on those wait lists. Most of the other organs that are transplanted are in the 10,000 or less category. So this would be a dire need.
We're going to let Cory make his escape with 20 seconds left. Thank you.